Good afternoon and welcome to all. Today we are going to discuss about glyoxylate cycle. So let's go through the glyoxylate cycle. Introduction. It is a variation of tricarboxylic acid cycle or TCA cycle, and it is an anabolic pathway, which means the synthesizing pathway occurring in plants, bacteria, protists and fungi. The glyoxylate cycle centers on the conversion of acetyl coenzyme A to succinate. For what? For the synthesis of carbohydrates. In microorganisms, the glyoxylate cycle allows the cells to utilize two carbons, such as acetate, to satisfy cellular carbon requirements when simple sugars such as glucose or fructose are not available. The cycle is generally assumed to be absent in animals, with the exception of nematodes at the early stages of embryogenesis. Plants as well as some algae and bacteria can use acetate as the carbon source for the production of carbon compounds. Plants and bacteria employ a modification of the TCA cycle, and it is known as glyoxylate cycle, and it produces 4-carbon dicarboxylic acid from 2-carbon acetate units. The glyoxylate cycle bypasses the 2-oxidative decarboxylation reaction taking place in the TCA cycle, and they directly converts isocitrate through isocitrate lyase and malate synthase into malate and succinate. Similarities with the TCA cycle. The glyoxylate cycle, first of all, utilizes 5 of the 8 enzymes associated with the tricarboxylic acid cycle. First one, citrate synthase, then aconitase, succinate dehydrogenase, fumarase and malate dehydrogenase. So these are the first 5 enzymes associated with the TCA cycle, which the glyoxylate cycle utilizes. The two cycles differ in that in the glyoxylate cycle, isocitrate is converted into glyoxylate and succinate by the enzyme isocitrate lyase instead of into a alpha ketoglutarate. And it bypasses the decarboxylation steps that takes place in TCA cycle or citric acid cycle, allowing the simple carbon compounds to be used in the later synthesis of it is used in the later synthesis of macromolecules including glucose. So this may allows the simple carbon compounds to be used in the later synthesis of macromolecules, that means big molecules including glucose. Glyoxylate is subsequently combined with acetyl-CoA to produce malate, and it is catalyzed by malate synthase. Then malate is also formed in parallel from succinate by the action of succinate dehydrogenase enzyme and fumarase. Next is about inhibition of glyoxylate cycle. Due to the central role of glyoxylate cycle in the metabolism of the pathogenic species including fungi and bacteria, examples of the glyoxylate cycle are current inhibition targets for the treatment of disease. Most reported inhibitors of glyoxylate cycle target the first enzyme of the cycle, and inhibitors were reported for Candida albicans for the potent use as antifungal agents. The mycobacterial glyoxylate cycle, it is also targeted for the potential treatments necessary. So the mycobacterial glyoxylate cycle is being targeted for the potential treatment of tuberculosis. So this is the synthesis. First of all, acetyl pyruvate, which occurs as a result of the intermediates pyruvate, which may enter the TCA cycle. It is converted to acetyl coenzyme A plus water, which gives acetyl CoA and then water. Citric acid, the citrate and this acetyl CoA can be converted to citrate with the help of citrate synthase. Citrate can be converted to citric acid. Citrate, citric acid or citrate. And then this citrate can be converted to aconitase. Converted with the help of the enzyme aconitase to isocitrate. Then this isocitrate can be converted to succinate, or the isocitrate enzyme converted into glyoxylate. Next is glyoxylate is the key metabolite. So this formation of this glyoxylate with the help of the enzyme malate synthase, this may result in the synthesis of malate and the carbon dioxide is released or the decarboxylation occurs. And the malate, which can be again converted to oxaloacetate by the Cook NAD plus, and which can be converted to the intermediates NADH plus H plus. Then coming the oxaloacetate again, which may combine with acetyl coenzyme A plus water to form a sulfur. This oxaloacetate, which again combined with acetyl coenzyme A with the presence of the enzyme citrate synthase forms citrate. So the cycle is repeated. So I am telling the steps in glyoxylate cycle in brief. The glyoxylate cycle, like citric acid cycle, it begins with what? Condensation of acetyl CoA and oxaloacetate to form citrate, which is then isomerized to, it can be isomerized to isocitrate. Instead of being decarboxylated, this isocitrate, which can be cleaved by isocitrate lyase into succinate and glyoxylate. The subsequent steps regenerate oxaloacetate from the glyoxylate. Acetyl CoA condenses with glyoxylate to form malate in a reaction catalyzed by malate synthase, which resembles citrate synthase. Finally, the malate is oxidized to oxaloacetate as in the citric acid cycle. So here are the steps involved in the citric acid cycle. 2 acetyl coenzyme A plus intermediate NAD plus water gives succinate plus 2 coenzyme A plus NADH plus 2H plus. The glyoxylate cycle allows the plant and some microorganisms to grow on acetate because the cycle bypasses the decarboxylation steps of the citric acid cycle. The enzyme that permits the conversion of acetate into succinate. So what happens here? That is, enzyme that permits the conversion of acetate into succinate are nothing but isocitrate lyase and malate synthase converts this acetate into succinate. Bacteria that can synthesize acetyl CoA from the acetate and coenzyme A by an ATP-driven reaction, which is catalyzed by acetyl coenzyme A synthase. So then the next is pyrophosphate formed, it is then hydrolyzed to octophosphate. So the equivalence of two compounds having high phosphoryl transfer potential are consumed in the acid activation of acetate. So that's all about glyoxylate cycle. Next coming is about substrate level phosphorylation. The ATP synthesis in prokaryotes. The objective of the catabolic pathway, catabolic means breaking down, to make ATP, to transfer either chemical energy or electromagnetic energy into the shell contained with the high energy bonds of ATP. The cells fundamentally can produce ATPs in two ways: substrate level phosphorylation and electron transport phosphorylation. These are the two methods which the fungi, the protein produced fungi is in table. So here the cells fundamentally produce ATP in two ways, mainly through substrate level phosphorylation. Another is electron transport phosphorylation. And we are familiar with electron transport phosphorylation, which we know as electron transport chain. We have heard the term substrate level phosphorylation, isn't it? So we can discuss. Here, the substrate level phosphorylation is the simplest, oldest and least evolved way to make ATP. In a substrate level phosphorylation, ATP is made during the conversion of an organic molecule from one form to another. Conversion of organic molecule to one form or from one form to another form. And the energy released during this conversion, it is partially conserved during the synthesis of high energy bond of ATP. And also this substrate level phosphorylation occurs during the fermentation and respiration, and even during some lithotrophic species, lithotrophic transformations of inorganic substances. So this is the picture of substrate level phosphorylation, three of them. First one taking place in glycolysis, 1,3-diphosphoglyceric acid converted to ADP. At that time 3-phosphoglyceric acid is produced. Then 3-phosphopyruvic acid, they also disturbed. So sorry, the phosphoenol pyruvic acid, which is also the step in the glycolysis. This it can be converted to pyruvic acid by the synthesis of ATP molecule from the substrate itself. Here the substrate is phosphoenol pyruvate. So the synthesis of this ATP molecule utilizing the organic substrate is called substrate level phosphorylation. Third one, that is acetyl coenzyme A, which can also be converted to acetic acid with the help of the synthesis of the ATP, which may occur during this period. Acetyl coenzyme A to acetic acid. So again we are describing that three examples. Both examples of substrate level phosphorylation, both A and B, are two-level substrate level phosphorylations that occur during the Embden-Meyerhof pathway, that means glycolysis, but they occur in all other fermentation pathways which have Embden-Meyerhof component. Third one, C. It is a substrate level phosphorylation, and those are found in the Clostridium and Bifidobacterium. Both of them are anaerobes. These are two anaerobic bacteria who learned how to make one more ATP from the glycolysis beyond the formation of pyruvate. So these are all about glyoxylate cycle and substrate level phosphorylation. Thank you.